name's Ben, and I'm a grad student here at K-State. In our lab, we study small mammals. You may not see these mammals very often, but they're all around us, and they're very important for the ecosystem. They provide a lot of super important functions, such as dispersing seeds for plants, as well as acting as prey species for animals that you do see, like snakes and hawks. And so in order to study them, we do a lot of work in here, but we also have to do a lot of work outside. We try to answer questions about which species occur where, how big the populations are, how these species are related to each other, if there's any distinct genetic groups within these species, and things along those lines. So, with that, let's start our lab tour, but for that, we have to go outside. Konza Biological Station is located just south of Manhattan and has been used for ecological research studies since 1971. It is located on 3,487 hectares of native tallgrass prairie. We call it native because it has never been plowed due to the shallow limestone soils which make it unsuitable for planting crops. Because it was never converted to cropland, it is a very natural ecosystem and still has almost all of the plants and animals that historically live in tallgrass prairie. This makes it useful for scientists doing all kinds of research. Much of the research going on at Konza involves looking at the effects of fire and grazing, two very important things for a prairie. Konza is split up into multiple parts, or treatments. First, half of the treatments are grazed, either by cattle or bison, which there used to be millions of and which used to graze prairies all over Kansas.
What is a treat for a bison? This one's coming right up.
The animals that we work with today are going to be a lot smaller than these. Second, these areas are burned at different frequencies. Some every year, some every 2 years, some 4, and some only every 20 years. The fire and grazing treatments are combined with each other. So, each of these fire treatments has at least one version that is grazed by bison, one or more grazed by cattle, and at least one that is not grazed. Based on this, you can see the importance of fire and grazing for the tallgrass prairie. Without these forces, the prairie would turn into forest. This is because after burning, prairie grasses grow back quickly, but woody shrubs and trees can't grow back as fast. After a burn, the land looks dark and sooty, but very quickly turns bright green as the grasses start to grow back. In these overhead shots of Konza, see if you can identify which areas have been recently burned and which areas are already starting to grow back. In my lab at K-State, we study small mammals, and there are many species of them living at Konza. Because of the different fire and grazing treatments, there are a variety of habitat types for animals to choose from. There are many areas of tallgrass prairie which dominate in areas that are burned frequently, and also areas with more shrubs and trees which take over in areas that are not burned very often. So small mammals have a variety of habitats to choose from. On this map of the country, you can see the location of Konza indicated by the green dot. Because of the presence of both prairie and woodland habitats, many small mammals are able to live here. First, I'll go through the ones that prefer the prairie. First, we have the deer mouse, which is regularly found at Konza and has one of the widest ranges across North America. As the name suggests, the prairie vole also prefers the prairie. It may look like a mouse, but it can be distinguished by its smaller eyes and ears and darker fur color. Next is the harvest mouse. These mice are so small they can climb up grasses and balance at the top, like in this picture. The pocket mouse can also be found in prairies. It gets its name from its external cheek pouches, which it uses to carry around seeds and other goodies. Last for the prairie, we have the 13-lined ground squirrel. Its scientific name, Ecdytomys tridecumlineatus, is one of my favorites because tridecumlineatus literally means 13, tridecum, lined, lineatus. Up next, we have the species that prefer the forest habitat. First, the eastern wood rat, which is pretty common at Konza. It is also referred to as a pack rat because of its tendency to build large nests out of virtually any material it can find. As makes sense based on the name, the woodland vole can be found in woodlands. They look very similar to the prairie vole, but can be told apart based on fur coloring and the habitat they are found in. Next is the white-footed mouse, which is very common at Konza. This mouse looks very similar to the deer mouse, but there are a few nifty tricks to tell them apart, which I'll explain later. Last for the forest is the fox squirrel, which you've likely seen in Manhattan at parks or basically anywhere there are trees. They get their name from their reddish-brown color and big, bushy foxtail. Besides prairies and woodlands, the other main habitat at Konza is by the creeks and around other damp ground. The jumping mouse prefers these wetter areas. It gets its name from its big hind feet, which it uses to jump distances of up to 3 feet, more than 10 times its body length. Next is the short-tailed shrew. It looks a bit like a big mouse, but it is actually more closely related to hedgehogs than to mice. Its diet is made up of insects, worms, and other small invertebrates. Last up is the least shrew. This is one of the smallest mammals in the world, measuring only up to 3 inches fully grown. Like the short-tailed shrew, it eats insects and whatever else it can find. There's one more small mammal to talk about. The cotton rat, which is perhaps the most common small mammal found at Konza because it isn't picky about its habitat. It can be found in prairies, woodlands, and around creeks. In this video, I'll be setting traps at the Holbert plots, which are a miniature version of the rest of Konza. All of the combinations of burn treatments are represented in small little plots here to demonstrate what happens to the prairie depending on how often it is burned. In the distance, you can see a plot that is full of trees. That's one of the plots that's only burned every 20 years. In front of that plot, you can see 2 plots that are made up mostly of grasses. These plots are burned every 1 or 2 years. This area is where we're going to set traps today, because we can easily set traps in a variety of habitats here. To study small mammals, we have to find them first. The easiest way to do this is to set traps for them. Right now I'm setting Sherman traps, and so they fold up nicely like this, so you can carry a bunch of them around. We've got 40 of them in here, and so the way they work, they just fold out nicely like this. You can put down this little part in the front and it'll catch on a lever here, and then if something runs in there, there's a little trigger at the back so that when it gets hit, it closes, and so it'll catch a nice little mouse or rat, something that size. When setting traps, I always do it in the same general way so that I know how to find them later.
The way we normally do this is we kind of put two of them in a station. So I'm going to take the first one here. I'm going to put some bait in it. So now we've got some bait in there. We've got it nice and open so that an animal can run in there. And you want to try to move some of the grass out of the way, get it onto the ground where the actual mammals are going to be. And once it's down there, you just make sure it's open. And that's our first one. Station. That just kind of makes them easier to find once we go back later to try to find them again. So here we go. And we'll just go right over here on the other side, move the grass out of the way. All right, there we go.
With the first two traps set, I move on to start setting the rest of the trap line. Basically, I set them in a long, straight line made up of multiple stations. Each station has two traps, which I set with the open door facing the middle so I can check them easily. Once I've set the two traps at a station, I take 10 steps and then set the next station. Usually I set out around 40 to 50 traps in a single trap line, because the more traps you set, the more likely you are to catch something. I'll leave the traps out overnight and come back to check them the next morning. Once we catch some animals, we have to decide what to do with them. Sometimes we keep the specimens we catch to put in scientific museums. I'll talk about this a bit more when we go back to the lab later. Today what I'm going to do instead is release whatever I catch back to where it came from, after I collect some data for each specimen. This is a method called capture, mark, recapture. In this method, I give each animal I catch an ear tag so I can identify it again later, and keep traps out for multiple days. By trapping over multiple days and recapturing animals that were caught earlier, we can learn important information about each species, like how big their home ranges are, how many individuals are in each population, how population sizes change over time, and which species coexist with each other. In addition to this long line of Sherman traps, I'm also going to set out some bigger traps.
This is called a cage trap, and it's, as you can tell, a lot bigger than the other one. And so this will kind of allow us to catch more medium-sized mammals, so things like raccoons and possums. And it basically works under the same principle. We have the live store here. This is it when it's set. And if something comes along and triggers this lever at the back, it'll close and catch it. So, nothing in this one. It's like nothing here either. feels like we got something. Yep, looks like there's a deer mouse in here. Alright, looks like this one's closed. Let's see what we got. Feels like there's something in there. Oh, there's a deer mouse in this one. Okay, here where we set all the traps. The first couple cage traps didn't get anything, but we're going to check this last one here to see if there's anything in this one. Oh, it looks like we got something. Today what we're going to do is a lot of measurements, and then we're going to ear check and look at the information. We want to, first of all, make sure we wear gloves when we handle them, and then we have our gear bag here that we're going to use to do all the things that we need to do with them. So first of all, we've got some plastic baggies. We're going to use these to get the animals out of the traps. So I'm going to start off small. We're going to start with one of these white-footed mice. The scientific name for these is Paramyscus leucopus. And so we're going to take it out of this trap. The way to do this is put the bag around the trap like this. We're going to open the door now, so the mouse has nowhere to go but out into this bag. So, once this door opens, there we go. It'll open like that. We turn it like this, and the mouse should just come on right out into our bag. Once we have the mouse in the bag, we can close it like this so make sure it doesn't go anywhere. We'll put it down here on the bed, and it'll kind of like to go into the corner. Once it does, we can put our hand on it. We kind of push it down gently, but firmly, that way that its mouth can't get around to bite us. I'm going to put my hand up here on its head. Once I'm pushing down on its head, it can't go anywhere. And then I'm going to scruff it. Some of you might have cats, and when you have baby kittens at home, a lot of times you can pick them up by the scruff of their neck. We're going to do the same thing with this mouse. So now I've got it in my hand by the scruff of its neck. It can't go anywhere. It can't turn around to bite me. And so now we can start to do some work with it. Alright, so what we want to do, first of all, is get some measurements. And so for that, we'll go back to the gear bag. In here, we've got our ruler. So there's a couple measurements that we want to get. The first one is going to be the tail measurement. So in order to do this, we put the ruler here, get it all the way back, pull the tail down, and just read what the measurement is. So the measurement here is 90 millimeters. In science, we always use the metric system, and for these measurements, we're always going to use millimeters. So the next measurement we're going to get is the hind foot. So I've got the foot here, press it to the ruler, get the back on the end of it, get the toes extended as far as they go and read the measurement for this. So this foot is 23 millimeters long. And this is kind of an interesting case. There's two kinds of mice that look very similar. They both look like this. We have the deer mouse and the white-footed mouse. The way to tell them apart, first of all, one of the best ways is based off of the hind foot measurement, which is what we just took. And so I just said that the measurement was 23 millimeters, and so based on that, we know that it's a white-footed mouse. If the measurement is above 21, it's a white-footed mouse. If it's below 21, it's a deer mouse. So that's one of the reasons why we take these measurements. Another way to tell these two species apart is their tail. So if we can see it, we get a good look. You see the bottom of the tail is kind of a lighter color, but not very white. If you look at the top, it's a little bit darker, but back and forth, there's not a lot of distinct difference between these two colors. If we had a deer mouse, you would see a very dark top tail, and on the bottom, it would be very bright white. And another way to look at that is if you put it down and you look at the top, if you can see white on either side of the tail, then you know that it's going to be a deer mouse. If you can't, like in this case, then we have a white-footed mouse. So that brings me to the last measurement. For the last measurement, we're going to measure the ear. And so this one can be a little tricky. We're going to be working around its head now, so we want to make sure that it can't get around to bite. But if we do it right, it won't be an issue. So first of all, I'm going to take the ear. They get a little feisty like that, but if you keep a good grip, they won't be able to do anything. I'm actually going to put it down and get a re-grip on it. So I just do what I did earlier, grab its head, and then I scruff it again, like that. Alright, to get the ear measurement, we're going to stick the ruler inside of its ear and pull it up to get the full measurement of its ear. So here, if you can see, that looks like 13 millimeters. Alright, so those are all the measurements we're going to get. The next thing we're going to do is try to figure out what sex this mouse is. So if we look here, we can see, first of all, what's going to make it pretty obvious is the presence of nipples. So we can see there's a couple here, and so that we know in this case it's going to be a female. And so as we go through the other ones, we'll kind of look at the differences. If we have a male, I'll show you the difference between a female and a male. And then before we let it go, we're going to do a couple more things. The first thing we're going to do is get our tools out to tag it. What we're going to do is use these little metal ear tags in order to clip it to its ear so that it won't fall off, and that if we re-catch this mouse, we'll know which individual it is, and we won't have to do all these measurements again. We'll just know that we caught the same one, and we'll be able to see where it traveled, see if it moved around a lot in between our trappings. So, in order to do this, we use one of these nifty little tools. We're going to put the ear tag inside of it. Like so. Once we have the ear tag here, we'll go over here. We're going to go down as deep as we can into the ear so that it's less likely for it to fall off. And then just press it in. Once we've done that, we'll open it up, and there we go. This mouse now has an ear tag on it. And so once we're ready to let it go, we'll let it go and we'll be able to identify it again later. Before we put it back in the trap, there's one last measurement we need to take. We want to get the weight of this mouse. And so for that, we have these handy little measuring tools called cassolas. And it's pretty simple. We're just going to attach it to the foot of the mouse and let it go. And it just gives us an estimation of the weight. So this mouse weighs 31 grams. Now that we have all of our measurements, we'll put it back in the trap for now so that we can go back later and release it where we caught it. So to do that, we'll just put it in here like this, close the trap, and release the foot. And now it's back in here, safe and sound. Good. So we have another white-footed mouse here. Again, remember we can look at the tail to tell. So we would know if it was a deer mouse because there'd be a lot more white here, and it's very large feet. So it's going to be a white-footed mouse. This one is a male. So we see the difference between this one and the last one I had. The last one had nipples that we could see, as well as the absence of a penis. This one has a penis, as you can see right here. And as well, it's a scrotal male. We can see that the testicles have descended down into the scrotum. And so we take this information to get an idea of the reproductive condition of these animals. And that's just another thing that we can use for our studies. Mice now. I'm going to move on to the other two species that we caught today are both rats. We have a cotton rat and a pack rat. These are both larger animals, and so to be safe, I'm going to exchange my plastic glove for one of these leather gloves in order to handle this animal. And there we go. Basically, very similar strategy, still scruffing it, just wearing a glove, just in case. These ones will hurt a little bit more if they bite you. So now, I'm going to show you the difference between a cotton rat and a pack rat. So as you can see here with the cotton rat, it's got this kind of black and white, like salt and pepper color to it. Whereas the pack rat, which we have in the cage trap here, is more of kind of a grayish, tannish color. It's a little bit bigger than the cotton rat, and it has this kind of longer, thicker tail. Next and last, we're gonna do the last species that we caught today, which is the pack rat. For this, I'm going with both leather gloves so I can reach in and grab it without it biting me. Now that we're done getting all of our measurements and information, it's time to release these guys back into the wild. When we put them back, we want to put them back in the habitat that they came from. And so these mice are often associated with the prairie habitat. So we're going to release them back here. And so the way to do this, you can just open up the trap, let it go. Alright, here we have some of our caught rats. These rats are usually kind of associated with prairie, shrubby areas, but they can use a lot of habitats. So we're going to release it into this area right here. So we're going to release the last thing we caught, the pack rat. These rats like wooded areas. We're going to release it back where we caught it, here by these trees. And the reason they like these wooded areas is because they build their little nests or middens out of the sticks and little pieces that they find around the trees. And then they actually pee on them, which then crystallizes their middens into a more solid structure. Using the small mammals we collect, we do a number of things in our lab back at K-State. Much of the research in our lab revolves around using DNA to study the relationships between and within species. DNA is the molecule that contains all the information about an animal and every other living thing. It has the instructions for how to build a whole organism. These instructions are different for each species and always slightly different for every individual. In order to study DNA, we have to extract it from the cells of the animal. We use many different chemicals and follow a number of detailed steps in order to do this. Here we can see a few of the chemicals we use on a daily basis. We usually extract the DNA from muscle samples, which we have to store in a very cold freezer.
By keeping these samples at the very cold temperature of negative 80 degrees Celsius, the samples don't degrade and we can keep them safely for years. Here you can see a tube with a little muscle sample in it. The end product of a DNA extraction is a clear liquid in a small tube like these. You can't see it, but tiny strands of DNA are floating around in these tubes. By studying small mammal DNA, we can understand how species are related to each other and if there are major differences within species. Sometimes there are big differences even between animals that look the same. Other work in our lab involves the parasites that live inside small mammals. Parasites are anything that make use of another organism's resources, to the detriment of that other organism. In the case of small mammals, we are interested in studying the small worms that live inside of them.
Hi, Kaylee.
Oh, hi, Ben.
What are you working on?
Right now I'm looking at the parasites of a shrew species from Alaska.
Ooh, how do you do that?
You take the guts from the shrews and you put them under a microscope, and then you look for anything that looks like a parasite.
So Kaylee, we study mammals. Why do we care about the parasites?
That's a great question, Ben. This project in particular, we're looking at shrews from the mainland in Alaska and shrews from a small island called St. Paul. And the mammal diversity on an island is a lot smaller. So we're looking at the endoparasites to see if that is also reflected, if there will be less species and genetic diversity on the island population.
All right, I found another person working in the lab. Let's see what she's up to. What are you working on, Lisa?
I'm entering data.
Entering data. So, I'll give you a little background. A lot of times with the work we do in the field is what we just showed you, where we collect, tag, and let them go back into the wild. But sometimes we collect whole specimens. And the reason we do this is to kind of get documentation of the biodiversity so that mammals that exist at this time and at these places. And this just helps for conservation to be able to say for sure that these things occurred in these places. And they can help with a wide range of other studies too. So one of the main things we do with this kind of work is we take the specimens and we keep their skins intact like this so we can see their morphology, so their color, as well as how big they are, and also see just the relationships between the same species at the same locations and between other locations. So here we have one of the rats that we caught earlier. It's of the same species. This is one of the cotton rats, and you can see that we're able to preserve it so that it keeps a lot of the morphology the same, so it looks like the same color and about the same size. We also have some cool examples of species that came from other places around Kansas. Here is a flying squirrel, which occur in eastern Kansas in woody areas. And you can see the tail has these hairs sticking off it to help it glide. And you can see the patagium, which is the wing-like structure that extends from the forefoot to the hindfoot. And when it extends its feet out, it's able to glide from tree to tree. So that's one example. We also have another kind of rat. This is called a kangaroo rat, and they occur out in western Kansas. And they have this cool orange color and this really long tail that helps them balance when they jump. And so by preserving these specimens, like I said, we're able to understand what species occur where and make comparisons between them. And in order to do this, what's also really helpful is to have really good data keeping. And so that's why we have these giant folders full of data, and that's one of the things that Lisa works on a lot. Would you be able to walk us through one of these data pages?
Yeah. So the first block here is where we take what the species is and we do the day we caught it, and this is just to help us know exactly where we caught it from so we can go back and put in this information later. So this is all of the information about where we got it. This is the state, the county, some specific area locations, and then we have the GPS coordinates down right here. And then after we do that, when we're in the field, we do the exact measurements of it. So we've got the total, the tail, the hind foot, the ear, and the weight, and that's all done by hand, and then we write it down on this page. We figure out what sex they are, if they're a male or a female, and then we write down their reproductive condition. We say if they're a baby or a subadult or a full-grown adult. And then we'll also check them for ectoparasites like ticks and fleas and endoparasites like tapeworms. And after we do that, we pass it on to somebody else and they'll take out the heart, the kidney, the lungs, and the spleen, the liver, the muscle tissue, and then sometimes we also take out the very end of the intestine. And then this specimen specifically, you can see that it has nematodes, which is another endoparasite that's like a little worm in the stomach. And then it also had a flea. It had 2 fleas actually.
So these skins, as well as the other things that we collect that Lisa was talking about, can be used to analyze a wide range of things. We can do genetics, we can investigate their diet, we can investigate life history, reproductive behaviors, as well as investigate their conservation status.